So we are going to see some applications of these optogenetics, which is are new tools to study real-time single-cell analysis. And so I think Alan explained very well why we are interested, or many of you know already why we are interested in nanotechnology. And then we are going to see these new tools to single-cell analysis that has been defined as optogenetics. And then we will go through some applications, in particular of the surface-enhanced Raman spectroscopy. And I'm just actually sharing with you what I've been reading in the literature in the past few months. So we'll see together some of the applications. So again, why we need to, we are interested in understanding what's going on in a cell that is exposed to ionizing radiation or any other kind of stress. So we use nanotechnology, something that can really go into our cell. And for example, these probes can be coated with, for example, silver and gold, and this would be the sensor that will help you understand what's going on, to analyze what's going on in a cell. So already 40 years ago, microbiologists knew that some microorganisms produce light-gated proteins that regulate ion flows. So, and it was in the 70s that this protein, the bacteriorhodopsin, was discovered to be a mediator of, was an ion pump. So in the past 30, 20 years, a whole family of these rhodopsins, or anyway light-activated protein, has been discovered, and they have been used at the beginning in neuroscience because just with light you could turn on or off neurons. So optogenetics started with neuroscience because those are excitable cells that could be switched on and off depending on the light. So for example, you could stimulate the cell, the neurons, just let's say with this red light. Or for example, using blue light and actually making use of proteins that are sensitive to blue light, maybe you can excite, in this case, nerve cells. Or in contrast, using, like in this case, yellow light, you can inhibit the transmission of signal between nerve cells. But as you can imagine, these can be applied to excitable cells like neuron or cardiac cells. But if you can just change the membrane potential of your cells, optogenetics can be applied also to any kind of cells. It doesn't have to be just excitable cells. So what are some optogenetic tools that they are using now in biology? So they make use of photosensitive proteins. For example, there is a protein, opsin, that belongs to those rhodopsin family, which is a transmembrane protein that is activated by retinal. There is a chromophore that is present in most mammalian cells. So when the light comes, this chromophore is activated, And then it gives rise, activates opsin that give rise to a specific cellular signal. For example, there are light-activated adenylyl cyclase that many of you might know are part of the G protein signaling cascade, which is one of the major signaling cascades that our cells use for many endpoints. And for example, the G protein signaling cascade will give rise to the release of cyclic AMP, which is a second messenger, very important for many signaling pathways. Another type of photosensitive protein are those channel or dropsin. So those are channels that carry these chromophore that once is activated, let the channel to be open, or in contrast, you can choose to close the channel. And for example, we are talking about ion channels. For example, you can decide to open or close calcium channel, and just switching on or off a specific incident light. So one thing that came up in my mind is that, for example, calcium can modulate the opening and closing of gap junction that are one of the major players in ionizing radiation induced bystander effect, for example. So you might use this kind of tool, so light, to open and close specific connexin channels, and then see if you get more or less bystander effect, depending on the cell that you have, cell density. So you can just play and design any experiment as such. Another photosensitive protein that has been used in optogenetics are those chimeric proteins like OptoXRS that are made by a light absorbing domain plus a protein effector domain. So as in this case, for the OptoXRS, you have light incident on the opsin that gives rise to the activation of the G protein mediated signaling cascade. And then there are like also this LOV2 plus the chromophore flavin that seems to be implicated in cell movement. So I was thinking maybe something like that might be used just with the turning on and off a specific incident light. You might maybe study cell invasiveness. So that could be an application. Or another tool in optogenetics might be the activation of light-sensitive enzymes, such as some that are implicated in the activation in the formation of actin filaments. And then again, you can think about many different applications about any stressor, so ionizing radiation, for example, and how if you can control actin filament formation depending on the stress that you have. And then another thing that I found very interesting is the use of optogenetics to have photosensitive release of caged ligands. So for example, you have some ligands that are closed they are caged in maybe nanoparticles somehow, and with the light you can somehow release this ligand that then can be delivered. Now, the beauty, I think, of this delivery system is that it can work with any antibody-antigen couple, and so any ligand-receptor pair. And then if you can think about, you can decide, maybe just turning on and off, switching on and off light, where you want to release your compound. So for example, you can think of release a certain compound, for example, antioxidant, after a stress. So maybe after a low dose of ionizing radiation. That could be another nice experiment to look at. And then one of the methods that with Alan and in the lab we are interested in looking at is a specific method in optogenetics, the surface enhanced Raman scattering nanoneedles. So those are nanoprobes that are coated with gold and silver nanoparticles. As I told you before, those will work as a sensor. So why? Raman spectroscopy is based on the scattering of photons by molecular bonds. So you have light that it's traverse your molecule, your compound, anyway, your target. And then upon interaction, the photon is scattered and it can lose part of its energy or can gain some energy. And if you look at this loss and gain of energy with a spectrometer, this loss and gain, they appear as lines. So you have a Raman scattering, which is what they say is stroke shifted when it loses energy, or anti-stroke shifted when it gains energy. So the idea is that the scattering of the incident photon causes changes in the vibration of the molecule that it's receiving the light, so of that substance under investigation. So again, in view that with a spectrometer, those vibrational changes appears as lines. So the bottom line of the story is that each molecule has its own Raman spectrum. So by investigating, by inserting your nanoparticle, and at the end you have your spectrometer, Raman spectrometer, you know that for each line you can tell what's inside that cell, for example, that nucleus. Protein has the Raman spectrum. DNA has another Raman spectrum. So you can start to thinking of what you can do. So again, our special target would be the cell. So we have an incident light and Raman stroke and anti-stroke light that is then conveyed to a detector. And what you get are many applications that you can do. So if you think about, you insert your nanoneedle in a cell, and if you think that if each component, each compound has its own Raman spectrum, at the end you end up with having a kind of nanomass spectrometer that you have exactly in a single cell or in a single compartment. So, for example, you can, differentiate the molecules that are, let's say you put your needle inside the nucleus of a cell and you can differentiate the molecule before and after a specific stress, for example, ionizing radiation. Or you can measure change in membrane potential in excitable and non-excitable cells. So now let's go through some of the literature that I'm reading about what people actually is doing with optogenetics. And you are very welcome to stop me, and if you're thinking of application that you might want to use, or please let's talk about together. So for example, those are HeLa cells, and the nano needle is inserted either in the nucleus or in the cytoplasm. So this is the blue line for what you get at the spectrometer when the needle is inserted in the nucleus. The red one is when the needle is inserted in the cytoplasm, and then the black one is when the needle itself before insertion. So if you look at the intensity of these spikes depending on the function of the Raman shift, what's the first difference that you see between the blue and the red spectra? Is there any? Sorry.
Shift starts early when the needle is in the nucleus.
The shift starts early. We are about 60 nanometer. Something else?
Intensity and the wavelengths.
The intensity of the bands. So what can we read at a spectrometer at 60 nanometer? Sorry.
600 is normally used for proteins, amino acids.
DNA. And as you can expect, there is no DNA in the cytoplasm. So you don't have signal around 60 nanometer because there is no DNA. So again, yes. I'm sorry, you wanted to ask something? No, okay, all right, sorry.
DNA is mainly absorbed at 260 to 2.
For the Raman, well,
This is Raman shift.
this is the Raman
shift.
shift.
Right.
Yeah, this is the Raman shift. Yes. So an application that you can think about is that you can analyze compounds in different compartments of the cell. And also, for example, you might decide even to micro-inject something in a specific compartment. So micro-inject, for example, an anti-tumor drug just in the nucleus or just in the cytoplasm or cytokines, whatever. Yeah, there are many things that one can do. And then you can also analyze the response to perturbation in the microenvironment. For example, these people, they inserted the micropipette in the cytoplasm before potassium chlorine treatment, and then they look at the Raman shift time after this KCl treatment. So you can really choose how to perturb your microenvironment and see what are the difference in the compound inside the cytoplasm or the nucleus. So another application would be exploit the fact that, as you know, some complexes, macromolecular complexes, concentrate in are divided in compartments between a cell. For example, ribosome synthesis occurs in the nucleoli, or the DNA is packaged, is folded in eurochromatin or in very highly dense packed heterochromatin. So here are an example. These are examples of HeLa cells, and they were analyzed. The nanosensor was inserted in different compartments of the cell, And they were able to evaluate, depending on where the sensor was, they were able to evaluate, because as I told you, each compound has its Raman spectrum. They were able to evaluate the amount of protein, DNA, RNA, and lipids, depending on the compartment. So they put their needle in the centrometric heterochromatin, and as you can see, you have a lot of proteins, RNA, DNA, and lipids. And those was done in live cells. But then if you go to the extra chromosomal region in mitotic cell, there is no DNA. They couldn't detect. Well, of course, they set threshold of DNA level, RNA, lipid level, so there was no DNA in the extra chromosomal region, as you can expect. Instead, in the nucleoli, they could detect a very high amount of protein and RNAs and very little amount of DNA and lipids. And then when they put their needle in mitotic chromosomes, they could see, again, a lot of proteins, a very high level of RNA and DNA that you can see, for example, here is 45, here there is nothing, here is 14, here also there is a very little. So in mitotic chromosomes, there is a lot of DNA. And then I still don't know why there is very, very little need for lipids in mitotic chromosomes. I don't know. So I found just very curious. Yes?
Can you look for mitochondrial DNA, Raman?
Oh, I don't know. I don't know if it might be. I should just look in the literature. I didn't see yet analysis for mitochondria, but yeah, it's something that one can definitely think of. And then just thinking how you can use such a technology. So, for example, one question would be, do those Raman spectra in specific subnuclear domains change depending on the radiation dose or the radiation linear energy transfer? So, if something changed depending on these variables, do the cells return to the normal condition? So you can study the kinetics of many things. And then another application that I was reading about is optogenetics to differentiate in DNA packaging. And this paper relates to a different DNA packaging in sperm cells, and it seems that differential packaging, in particular in human sperm cells, influence the viability of these sperm cells. So just looking at the ratio between the DNA in sperm cells is rich in proteamines. So just looking at the ratio of those proteins that are bound to the DNA, they could evaluate the strength of the DNA packaging in these cells and then make predictions about the viability of human sperm cells. Yes?
I think this technique will be very powerful even to study the different stages of the cell cycle. In the different stages of cell cycle, as you know, the DNA is bound with different types of proteins. During replication, those proteins will be bound.
Fantastic, yeah.
So one can really find out about the phase of the cell cycle?
I think, yeah, if you synchronize, you know, you have your set of cells and synchronize them in different phases, and then you probe, I bet you will find differences in Raman spectra.
I mean, Marcelo pointed out the limitations of H2AX, whether the cells are in this phase or the other phase. Instead of looking those different probes, different parameters, one can simply do this Raman spectra.
Yes, yes, definitely. And then,
I think that the difference between the X and Y variants, but there's a big difference. The big X chromosome and a tiny little Y chromosome.
yeah, yeah, that's also, yeah, we were trying to think, should we, I mean, can Raman spectra be useful to evaluate, to differentiate between chromosomes? So this is something really, really open. I found just a paper that was published in 2006. So can we really go from the DNA packaging to the actual chromosomes? So these people, they use laser tweezers with Raman spectroscopy, and they claim that they could differentiate, depending on the Raman spectra, between chromosome 1, 2, and 3. Well, actually, I really looked close to those lines and spectra. I cannot see much of a difference, but they evaluated the Raman spectrum of these chromosome 1, 2, and 3 from 6 donors over 12 days, and they evaluated they did a generalized discriminate analysis of such. So they say that they were able all the time to have consistent Raman spectra for chromosome 1, consistent Raman spectra for chromosome 2 and 3. And they did something similar analysis. They evaluated Raman spectrum of chromosome, again 1, 2, or 3, of a single individual over 6 days. But again, this is a paper that came out in 2006, had a couple of comments, so, and eventually those are just chromosomes 1, 2, and 3. But anyway, this at least opens the possibility to use this technology to also differentiate between a chromosome without any kind of staining or any time-consuming assay. And of course, you can, I don't know, just let your imagination go and think about different applications of this technique. And many different users might be willing to use this technique for microanalytical chemistry, forensic studies, microbiology, and you can apply, actually people applied optogenetics to cells, C. elegans, mice, rats. So it's really open, and that's it.